favorite fuck squats.
Honestly.
Oh my god, also
Either
look, I
fear
got
faculty
new
or students.
Isn't that amazing? Literally so cute.
You're so cute and you
I know!
Yeah.
Did I tell you I got a new cat? He's a month. He's just reached the 1 month in my
The
household.
glue screen protector.
What's
I did buy it.
His name is Percival or Percy. He's a very refined fellow. He's 23 pounds.
20 pounds?
23?
23.
That's a choice.
Yeah. Right. Yeah, this is everyone. Whoa.
I guess I'm supposed to
Yeah.
Henry.
Oh,
okay.
We're doing introductions, yeah.
Waiting for your thing to go off.
Oh, I can do that.
Okay, hi everyone. I guess this is our first GSI Work in Progress talk of this year and maybe ever, or since I've joined.
Maybe the last.
Maybe the last. So the presenter today is Henry Sun. Henry is a third year in Finn Hawkins' lab, and his talk is titled Airway Stem Cell Engraftment for Early Intervention in Chronic Airway Disease. And some fun facts about Henry: you saw his new cat that he has fun doting on, Percy. Henry is also a local coffee shop peruser.
That's all of me. That's everything. All right. Cool. Hi everyone. Thank you for being here. Like I said, I'm Henry. I'm a third-year PhD student in Hawkins' lab as part of the Center for Regenerative Medicine, or the CREF, as we like to call it. And we're focused on using iPSCs for regenerative medicines. But sadly, my talk will not focus on iPSCs. But if you want to talk to me about them, they are an important part of this vision that we're going to talk about. But yeah, come find me if you're curious about that. So my talk is about my project, Airway Stem Cell Engraftment for Early Intervention in Chronic Airway Diseases. Breaking that down a little bit in different parts. I first want to just orientate you to your lungs, your respiratory systems. So we can broadly break it up into our airways, which serves the role of filtering and trapping particulates and pathogens as air travels down to your lungs, where in your alveoli, gas exchange occurs. These are two different parts, and it's important to differentiate these because they have unique cell types, morphologies, and functions. In my lab, we're particularly focused on the airway, and this particular little cell here, the basal cell, which we'll get back to, is the resident stem cell of the airway. I just want to orientate you to this difference, and when I say airway, this is what I mean. Also importantly, we're talking about airway disease, so I just want to talk to you about the big picture. So chronic respiratory diseases, which include chronic airway diseases, affect a lot of people, almost 500 million people globally. And one of the main contributors is chronic obstructive pulmonary disorder, COPD, which has both a alveoli and a airway manifestation phenotype. And unfortunately for people who have COPD, and it's characterized by inflammatory state in both these components, there is no cure outside of a lung transplant. So you get this at some point in your life, and unfortunately then progressively over the course of your life, you will experience respiratory decline, and you might potentially end up needing a lung transplant. You can see for lung transplants, about a third of recipients have cases of chronic obstructive pulmonary disorder as a reason for needing a lung transplant. So, lung transplants, great. They can cure, they can restore respiratory function, but they have a lot of limitations that we need to address. You can probably imagine they have eligibility requirements that disqualify a lot of people who need them. Easily, you guys can imagine that the demand for compatible, healthy donor organs far outstrips supply. And despite all of that, they also have fairly poor survival outcomes. So the 5-year median survival for a lung transplant is about 5 years, which is much worse than most other or all other solid organ transplants. And that's a really big part that limits how we approach treating chronic airway disease. So to give you kind of like the experience in my head of what a patient might experience. The pipeline, a quick pipeline is, they around the age of 30 or 40, they experience difficulty breathing, so they go to their doctor does tests and maybe does some genetic genotyping and say, hey, you have a lung disease like COPD, maybe genetic cystic fibrosis or cardiac dyskinesia. We can't cure it, but here are some supportive options like bronchodilators or kind of mucus rinse practices. We'll see you when you become really sick. And so then you would go back and you would live your life, try to be healthy, not smoke, including vapes. But over the course of your life, you will experience respiratory decline, and you're at elevated risk for experiencing end-stage lung disease, at which point you end up back in the hospital and you require a lung transplant to continue living a relative decent quality of life. But then even after that, as we've talked about, there are a lot of limitations following that. So our goal, our big vision with our research is to think about an early targeted option before the onset of lung disease and the onset of this irreversible lung damage. So for example, could we intervene at this stage with some treatment option? And so that's where we're going to approach cell-based therapies. And they've been used harnessing the regenerative capabilities of adult stem cells, most prominently, in many of you know, in the HSC field, both treat blood malignancies and in combination with gene-correcting techniques to treat genetic diseases like sickle cell disease and beta thalassemia. It's really incredible, and it's spurred a lot of research, companies, treatments, and a lot of really interesting research groups, including in the CREM, this is the Nutzel lab. So taking inspiration from this field that has proven the capability of this approach, we want to ask, can we apply a similar approach of grafting stem cells into the airways to treat chronic airway disease? And so, we talked about the basal cell before. I'd like to just come back to that as the resident stem cell of the airways. What this means functionally is that it is capable of multipotency, becoming different cell types, as well as indefinite self-renewal. And I will show you that first part in the seminal paper where this group first labeled these terminal cell types, these terminal cells, multiciliate cells that line your airway epithelium, and they brush mucus along. They express this ciliary protein, it's in brown. And then they use a laxity reporter to lineage label these keratin 5 positive basal cells, seen here in blue. And you can see over the course between 3 to 12 weeks, the transition, the retention of this reporter as these cells differentiate and colocalize with this terminal acetyltubular state, showing that basal cells become this acetyltubular cell, positive cell. And then this group did further quantification, also looking at another population of cells in the airway, the secretory cells, and they see that over time, percent of labeled basal cells decreases as the percent of lineage labeled ciliated and secretory cells increases. In this paper, they also show evidence of self-renewal, basically that these cells, they can serially passage. I think they did like up to 8 times, and they retain their capability to proliferate. I've included because it's just a table, it's kind of boring, but this is important definitions for stem cells for durable, grafted durable treatment. So, there's no questions there. Great, I've introduced you to the cell. I've introduced you to what we want to do with the cell. Now I want to ask, how can we study the engraftment of airway basal cells in the airway? We can't just go straight to the person, even though I sometimes kind of want to. So to do that, I'd like to introduce you to this murine model that we have. So to give you context, for a long time, the field didn't have a great way to study the biology of airway engraftment. There are some in vitro assays, there's some pretty bad in vivo models, but relatively recently, our group in the CREM, from the work of a MD PhD student in the Khan lab, has demonstrated the use of the murine trachea as a model for basal cell, airway basal cell engraftment. And the murine trachea is really interesting for this purpose because it anatomically is very similar to small human airways, has similar cell types, and for our purposes, it is much more easily accessible. So you can see here, in the bottom left, following airway preconditioning with a detergent solution, we get engraftment and engraftment of donor GFP positive cells, showing that these cells can adhere. And then with some IF quantification, again looking at this differentiated cell marker, acetyltubulin, you can see the co-localization of the red with the donor GFP, showing that these cells do end up becoming this differentiated cell type in this mouse model. So this is really interesting. This is the model that I'm going to be working with to explore some of the questions of engraftment. And this is like a nice picture, but usually it doesn't come out this nice. So here's like some examples of stuff I've done. So you just sometimes don't have anything. It's kind of sad. Sometimes you get some engraftment, but you've done the sectioning from the trachea kind of badly, so you kind of have all this blurry. And sometimes it's okay, but then you accidentally took a picture that does not include the entire thing. So all this to say is that these are my first 3, and I would like to say they're getting better, but maybe
So injury is always required for this?
That's a really good question. I think
And how do you standardize the injury?
that is, okay, two really good questions. The first one, I think that's currently the dogma in a lot of transplantation therapies where, for example, when you think HSC, right, they're usually an irritation of bone marrow niche to get rid of endogenous unhealthy cells. You gotta open up space for these new cells to engraft. So I think I would say yes to that question. Usually there is injury. We like to call it preconditioning, but injury, yes. Standardization of this injury is really important because essentially we might not be comparing apples to apples if the area that we're reconstituting is not treated the same. That's particularly difficult in this model to confirm. I'm doing some experiments right now where I'm trying to do staining of exposed basement lambda to quantify the extent of injury and the consistency throughout my experiments, but that's something where it's going to be hard to have a measurement of that in each experiment in the same animal that you do this. So that is a really good point, but I'm trying to look at that. Cool. Okay. So what are some questions that are important for us to ask and answer for translation of this approach to humans? But first to ferrets, but eventually to humans. So these are kind of the questions that are kind of currently guiding my project. First, we want to know how efficient our initial transplant is. I showed you, some reconstitution. I showed you some cells being, ending up there. We normally deliver 4 to 6 million cells, and we currently have some evidence that it's not a very efficient process. So very few cells actually make it in there in that process. Survive that process, want to proliferate, and so on. Okay, for our process, but if we want to scale up to a larger animal or to humans, it's a much larger area to cover. We want to know what the regenerative potential for a basal cell is, both endogenous basal cell, one that's just happily living there, and also one that we put in. You can imagine this is an interesting question for stem cell biologists, but also for us to know how many cells we might need to actually engraft successfully. There's also some really interesting questions there right now that I'm writing about clonality and how that impacts that compartment and how that might impact aging. You can ask more about that later, or please don't because it's really stressful. And then finally, the important question, how can we enhance engraftment efficiency? This is, these middle two, or this middle and this right option, I've just kind of have some thoughts, not quite too much work on it yet, but I'll show you a little bit of data on trying to quantify the initial transplant with some pilot experiments. But this one we're kind of thinking, we're trying to brainstorm, and also at the end, if anyone has any suggestions, please help me and let me know. So to answer this first question of how efficient is our initial delivery, we start with our hypothesis that less than 1% of our donor basal cells initially adhere. You'll maybe see why, but and additionally, this is something I haven't tested yet, but we hypothesize that the smaller set of these cells that adhere are the ones that actually contribute to area reconstitution. So there's heterogeneity going on there, some clonality going on there, and we'll talk about more about that in a second. So to test this, you might imagine that trying to track individual cell resolution, individual cells might be a challenging thing to do. So we collaborated with a group at Case Western where they have this nuclear, this diagram is wrong, yes, I'm sorry, but a nuclear reporter that is engineered to be super bright, and we got some trachea shipped over from them, we extracted primary mouse basal cells and expanded them for transplantation. Going into a little bit about how we do that. Oh, this is a picture of what they look like. Sorry, this is kind of hard to see, but you can see individual dots representing nuclei, so it's kind of cool. And this is just in culture. This is kind of how the transplantation model works. We would perform our preconditioning with 20 microliters of our detergent solution in anesthetized mice, give it 4 to 5 hours to recover from that process, and then we would transplant in our 2 to 3 million cells in this case. Normally, we would harvest tracheas to quantify the engraftment around like 2 weeks. But in my question here, I'm looking at the initial delivery. So I'm harvesting 1 day post, so it's 2 kind of busy days. And then I am sectioning, or I'm flaying open the tracheas and mounting them for either Nikon or focal imaging. And then I'm taking the tracheas afterwards to try to orthogonally validate my observations with flow cytometry. So that's kind of like the methods. Yeah.
Random question. With the detergent solution, do you notice like any phenotype? Do the mice start like coughing? Like, is it really irritating to their airways, or is it kind of just like a solution that you add and then it causes that
well,
injury?
apparently first thing, random thing I learned, mice can't cough, apparently. Really interesting. But we have seen through histology that this injury model is very severe and induces very significant sloughing of the airway epithelium. So the mice feel it for sure. They can't tell me the words,
Right. Okay.
but
Fair.
it's a severe model. And in some cases, if the injury goes too distally into the distal airways or into the lungs, we do notice airway or hemorrhaging in those areas. So it is quite severe. That's why we try to keep it to be proximal airways. Yeah. Sorry. Are you Owen?
Yes.
Okay.
You talked about the 4-hour recovery. Presumably in that time, there's a full scale response going on to the injury. This might affect how the transplantation happens, right? Yeah, so why 4 hours?
That's a really good question. Unfortunately, the bad answer I'm going to give you is what I've been told is that it worked in the model, so that's why we keep doing it. Otherwise, I'd have to prove other timing. We choose that time because we want to balance the time in which the niche is still open, but the turgid has cleared a little bit. Some of the chaos from the sloughing has subsided. That's a really good point that the immune cells must be signaling, especially significantly at that time point. Whether that's like helpful for us or detrimental, we don't know yet. But that's definitely a good question. Those are even also signals that I'm thinking of trying to upregulate in our donor cells beforehand to try to make them better at grafting, kind of like being ready for that injury niche. But those are just thoughts. Great. Okay. A couple pictures. Okay, so I got in trouble for this recently, but I'm going to say there are a lot of caveats to this data, and this is qualitative things only about these two tracheas with only an N of 2. Meant to do more, but expanding cells sometimes is difficult to go according to plan. So we quantify. It's kind of hard to see with the lights on. But with this nuclear signal, we get in these two tracheas between 1 to like 4,000 donor cells. You can kind of see in this close up each nuclear signal, and then I use cell profiling to try to quantify the nuclei here. The takeaway from these two tracheas that I cannot, will not overstate is that we have less than 1% of our 3 to 3 million donor cells being present at this time point. Caveats of this also include that we can't track if this will end up in reconstituting the entire airway because this is just that initial injury. We're doing some experiments that maybe try to look at that, but that's a really big caveat. So that's the imaging analysis. Again, try to orthogonally validate this. We do some flow cytometry. So up here is I'm just going to ignore these first 3 gates because it's just kind of our standard gates. We have a wild type control. You can see we have our negative reporter positive population, or negative reporter population. And down here in our actual sample that I pulled the 2 samples together, you can see that in our positive population we have very few cells. And when we do the math and calculate the number of cells in this population, it would represent, again, less than 1%, 0.1%. So this is just some preliminary evidence that this process may be very inefficient. As I've gotten told recently, is it representative? Unclear. I have to repeat this with much larger cohorts, which sucks because it takes a long time to expand the cells and the process kind of sucks. I have additional questions about if all these cells are seen here are contributing to repair, or if some of them are just kind of just along for the ride, they just stick, but then they just end up dying after this 24-hour time point. I'm planning on doing some Ki-67 stains to see if the cells are replicative. And I'm also kind of attempting a weird explant culture where I have a picture here of this trachea transplanted with both red and green cells to look at some clonality. But then I've chopped off a little piece of it and I'm trying to culture it in some 3D matrix gel to see if this will expand out and give me a sign of the reconstitution. This is like a random idea I had, so this might not work. The trachea is probably dying, so I will let you know how this goes. But also, this also sparks some really interesting questions that I've written about in my calls. Like, what is the distribution of the unique clones in this compartment of our donor-mated cells? This is a really important question for transplantation approaches because you might imagine if we have a very small population that actually engrafts, this could lead to a higher mutational burden or proliferative burden on those small population of cells, which might lead to higher potency risk. It might also lead to a more confined cell response because you only have so many cells. This is kind of something we see in the HSC field. We don't know if that's the thing here, but that's just a question I want to look at. And also, very importantly, for like mechanism, is there anything special about the small number of cells? If you guys have any ideas, that'd be great. Recently, I've been inspired in thinking about somatic mutations as driving perhaps some of either the expansion or the adhesion of these cells, and I'm planning on doing some whole genome sequencing to see if there's like an additional mutational burden, just generally or in specific oncogenes for the cell dying graft. But those are all kind of ideas that sparked writing my quals, which is going great. I just want to just say, have a month. It's going great. Great. Any questions about this section? I think this is like the data I was going to show, and then I'll talk about some plans.
Have you tried multiple types of donor cells, or what exactly are you?
That's a really good question. So in this case that I'm showing here, they come from a transgenic mouse that I believe is of a similar Black 6 background. We have looked at some mouse IBCs for transplants, but those are a little tricky because they're a 129 background, so we have to do the transplantation in compromised mice. Separate from that, we generally stick with wild-type mice, either labeled with the lentivirus or UBCGFP expressing Black 6 mouse. So a little bit on the string, but it's not a whole complement, which is good.
Yeah, that's really interesting because I was kind of thinking about extrapolating to human treatments and whether the vision was to have like iPS cells from a patient that would not be immunoreactive versus donor cells from some other source.
Ooh, okay, so I probably should put a slide in about that. But that would be the future vision. That would be the vision of the approach, where you could collect, let's say for someone with a CF mutation, their peripheral blood monocytes, reprogram those into iPSCs, and then differentiate those to these airway basal cells, or IBCs, iPSC-induced basal cells. And then from there, transplant those cells. So exactly to your point, you'd have an easy source of primary cells, epigenetically reset, and you wouldn't have to worry about immune rejection. So that is part of the grand vision. But right now we're staying within the mouse system because engraftment there just seems to work better. And so there's just a lot of basic biology questions about it that we want to answer. But that is like the final vision. That is I've done it, I've won a Nobel Prize. Yeah.
Super interesting. I guess I have a question about the engraftment. Maybe the efficiency is good. I mean, have you done sort of calculations of how much area you've cleared and how many of these basal cells would be ideal to populate that cleared area? I mean, maybe that's all the niche can tolerate. And then I guess the second part, maybe a suggestion. Have you ever tried putting in fewer cells? Because when there's a lot of cells, they may just prefer to stick to each other through like the lateral interactions of these cells versus from the basal side. Like if you just put in 1,000 cells, is the efficiency potentially better for the space?
To answer your second question, I have not, but from advice from the people who worked on this model before, they said that they screen lower amounts of cells, and once you go past, I think, 1 million cells, their signal just drops off a cliff. We do plan to just do that experiment, just have our own data for it, but that's just kind of why we haven't done it yet. And your first question was about kind of like quantifying the area and like normalizing that essentially.
Okay. What is the number of cells that would be ideal to engraft?
Oh, that's a really good question. So that's in a lot of ways we really don't know because it kind of might depend on the indication that we're trying to treat. We don't know. But for example, for something that is like cystic fibrosis, which you might be able to get away with having like a little patch of really active, ionocytes in one part of the trachea, the airway, you might not need the entire trachea transplanted. But for something where you need, like for example, mucociliary clearance, that beating action, you might need transplantation and engraftment to go along the entire trachea, or the airway. So it's kind of a tough question to answer, but it's a good question.
What is the normal distribution of these basal cells? Just how many are there per square, certain square unit of
That is a good question. I have tried to look into this, and it's surprisingly hard because ways that people would measure that would involve like dissociating them from tissue, which that kind of biases your readout. There's been some imaging approaches that just seem really raw. So I don't know, but I will try to have an answer in about a month for my boss.
It seems like it's an important, that's kind of following what Matt said, it's an important piece of knowledge that you want to try to have your system mimic.
Right, sure. I can give you estimates based on the digestions I've done from the harvested tracheas that I've taken, harvest large areas I've taken. But again, I'm not confident because our digestion protocol might not be perfect for isolating out, the ideal number of cells. That's a good question. Yeah.
The recipient mice, are they all immunocompromised?
In this case, we're going wild type into wild type, or sorry, black 6 into black 6. So no. In cases where in that published paper I mentioned, they did transplant mouse and human IBCs and human basal cells into immunocompromised mice.
So the rock paper was immunocompromised.
Sorry, the mob paper.
Oh, my immunocompromised. Okay.
Had some immunocompromised. Some of them were also wild type. Yeah.
But does that influence the recipient's reception of these transplantations? Depending on the immune state, and also have you considered variation in lung, mouth, microbiome status? Because there's a lot of microbes in the mouth that then connecting to the trachea, and your detergent is doing something, but do you know what happens in the microbiome, the airway microbiome when you do these detergent treatments?
Yeah.
These injuries.
That is a really good question about the microbiome. I do not know about that because again, we're inducing a sterile injury in this case. Again, if we induce maybe like a pathogenic injury, that might give us very, very different, like an influenza injury. That might give us a very different outcome result. I don't know if we can go with that approach, or just as an idea. In terms of like the immune background, that is almost certainly like a really important case where different signals there, like have TGF beta or Wnt signaling there, maybe not Wnt signaling, but like TGF beta or cytokine signaling that might apply a pressure to particular cell populations, or apply a advantage to the endogenous mesenchymal cells to help cells grow that we're missing. So that is very complicated, and eventually we do have plans to kind of try to do some sequencing and some inferred cell signaling between different compartments. But that is like a 60-year kind of yeah. Okay, so I'm just going to talk about some of the ideas for the other questions. So the other question we have is, what is the regenerative potential of a single basal cell? This is both in the context of an endogenous basal cell and of one that we transplant. You can imagine it's just an interesting biology question that we genuinely do not know. So, my current hypothesis, which is kind of, nondescript, is that a single basal cell clone is capable of giving rise to colonies comprising thousands of epithelial cells. It's just somewhere to start. Additionally, I hypothesize that transplanted basal cell will have similar regenerative capabilities to an endogenous one, which again, just somewhere to start. And the system that we're going to use to look at this, which I'm really excited about, is the confetti reporter system, which has been used to track cell lineages and clonal dynamics. So this is I think one of the coolest applications of it, where they looked at stem cell populations in intestinal crypts, and they found that each crypt is dominated by a single clone. They also apply this to other tissues. Just in brief, how it works. Within the Rosa locus, there is a transgene containing 4 fluorescent reporters, but only one of them is active at a time, and depending on how the recombination happens, a different one can be active. And it gives you this combination of 4 different colors. So you kind of get this nice little confetti that comes out. And the system has been used in the airway for airway basal cells, labeling specifically airway basal cells on a keratin 5 promoter. But this paper is a good paper, but this paper kind of did a bad job of using this system, because there's they only look for 2 colors, and I don't think they're labeling as they're in this case, or additive. But this is a system that I'm proposing to do. I'm currently breeding inducible confetti keratin 5 Cree ERT mice, which I'm calling concrete mice because confetti high. And particularly, I'm trying to do this with biallelic confetti expression. So the system I showed you before drives reporter expression off a single copy of the confetti transgene. I'm trying to do 2 because I'm assuming, I've read a paper where they do something similar and they get essentially 10 combinations of colors. So I'm hoping this will give me more clonal resolution to identify individual cells. It's kind of exciting. I don't know if it'll work, but that's kind of the exciting thing. My mice are pregnant, so I'm going to be a mouse cat. So it's exciting. Yeah. So I think that's what I have planned for this. And then the experiment I first plan to do, again, to answer this question of the regenerative potential of a single basal cell, first in a homozygous confetti mouse after inducing recombination. I'm planning on inducing an airway injury with our transplant injury with polydopamine. I'm going to harvest a group, and then I'm going to take the rest of the groups and let them recover over the course of a month, do another injury, and then repeat that, harvesting groups each time. And what I'm expecting to see is how these clonal populations might shift over time, and how one population, one color might come to dominate the entire population of the airway basal cell department. This idea of clonal dynamics is also really in my F right now, so I'm kind of excited about this. And again, this is the endogenous compartment. We can plan to apply a similar approach to, let's say, the potential of our donor of basal cells if we transplant it into confetti positive cells. So that's going to be the plan. Any suggestions, or if someone tells me this is a bad idea, would be appreciated.
These colors will be genetically identical, so there is no stochastic kind of selection of clonality, or there is an advantage. Is this going to be competition, or it's just purely stochastic?
Great question. In theory, it should be stochastic, but in actuality, there is a bias towards the labeling. I think the way they have this set up, like the first 8 are more common than the last 2 for some reason. I don't understand. The system doesn't go 10% each. In terms of the actual basal cells, we know from literature, both from transcriptomic data and protein expression data and functional data, that basal cells are not a homogeneous population. So there will be some selective advantages of certain clones. It won't be. I don't hypothesize it'll be driven off the reporter, but that is something that will be interesting to see. And this is where kind of looking at mutational burden, somatic mutation accumulation through whole genome sequencing might be really interesting. Great. And just to finish up. Some further questions again. If basal cell clonality becomes increasingly homogeneous, is that bad? We don't know, but there's some evidence in the HSC field from HSC transplantation that is associated with aging signature and reduced resilience. And in transplantation, it leads to higher likelihood of malignancies and poor outcomes. Again, you just have like a smaller population to respond to things. But again, we kind of want to know, for the purpose of making things better and also understanding what makes these cells special, and also can we transiently upregulate these pathways to enhance basal cell regeneration transiently, so that we don't make cancer. And so the idea is can we modulate some of these pathways that we identify as relevant in culture prior to transplantation with like an agonist or an mRNA treatment. And again, these are fun ideas that y'all get to think about and formulate my F. So it's going great. Great, and I'm just going to finish up with this quick last slide. It's just like the question of how can we improve it, because again, for our ultimate application in airway treatments. These are just some ideas I've kind of jotted down, and again, if anyone has suggestions, this is helpful. We could think of using a self-surface marker strategy to identify a population that's just inherently better at engrafting, maybe not yet understanding why. We know certain populations are more proliferative in culture and also in vivo, so like NGFR, DCAM mark those populations. We can think of a short boost of basal cell program in culture before transplantation, either through P63 YAPTAZ, downstream proliferation of stemness markers that I mentioned there. Some of these are components of Yamanaka factors also. We can try to temporarily boost survival and apoptosis with BCL2 or urokinase inhibition. And to the question of initial adhesion, we could try to maybe upregulate integrins or service markers. Personally, I'm choosing to avoid this option because we're kind of not set up as a lab to understand cell migration and attachment behaviors, and it also just seems personally a little less interesting. But those are ideas, and if anyone has suggestions, please throw them out. There are no bad ideas except my bad ideas. Okay, no ideas here. Please let me know. And yeah, that's my talk. Thank you to my lab, especially to Finn, to be a great mentor. To the Con Lab for all their help, all the Krem cores, the flow cytometry core, Shabita and Sabita, and all their collaborators. Any other questions? I know. Yes.
That's super interesting. I guess in a COPD patient, I think one of the problems with COPD is also like lung cancers. Mm-hmm. So do you have any worry? When I was looking at your little colony, it doesn't look so different from a tumor taking off. So if you put these cells into a more pro-cancerous niche, is there any concern they're not going to be basal cells, but will convert into a tumor?
Yeah, that is a really interesting question. Is that a concern? I think I would say yes. I would say our model in a mouse and over the course of a mouse lifespan isn't really set up to answer those questions. I think both looking at maybe if these small colonies are accumulating somatic mutations at a higher rate, especially in oncogenes, that might give you some indication of that. Yeah, I think the answer is just yes. I will say though, the way that the imaging happens, you don't kind of get the side view to see if there's like a squamous morphology to them. But again, that would be a concern. We transplant cancer, it's bad. Yes, Shabita.
I think Matt's question is really interesting in combination with your idea about giving them like a developmental boost before you transplant the cells. And I'm wondering if you can kind of drive development to a certain point such that they won't continue down the cancerous pathway until we pass that point.
So that is a good idea. The challenge there, and it's kind of like the problem with like stem cells and stemness, is that there's kind of an axis of like potency versus self-renewal that kind of contrasts, where if you go down that route and basically try to limit the proliferation of the cell population, it might engraft for a short term, like we know some hematopoietic progenitors do, but it might not persist over like a long period of time. And then the ones that do persist are different ones, and so kind of lost a long-term clone. So it's a good idea, but this is just challenging. Again, we know a lot about airway engraftment, especially in humans. I think there's like a couple clinical trials, not clinical trials, they're just anecdotes of scientists, I think in Asia, just injecting people with basal cells, and they got a little better. Literally the measurement was like, oh yeah, they breathe a little bit better. So we just really don't know, especially in humans. Yeah.
Still struck by the detergent washing procedure.
Yeah.
How, if you don't do any transplantation cells and you just measure gene expression in the trachea, you're just perfusing the trachea, not going all the way into the lungs, or how extreme is that detergent injury procedure? Because what I'm curious about is just if you did gene expression analysis, how reproducible, typical is the recovery period? Are some mice, mounting a massive inflammation response, and maybe those are instances where the transplantation doesn't work at all, and some where it's more subdued, and
Yes, that is a really excellent question. I will just generally say we don't know. One thing, so I'm kind of on the side of like trying to make engraftment better from the donor cell perspective. My lab mate, Yulin, is kind of on the other side of trying to have a more translatable injury model, injury model. So she's looking at ways of tampering down the endogenous basal cell compartment so that you can have a milder injury that might not be so inconsistent or very extreme. So kind of we're like on the both sides of the coin, and I think Finn's plan is we'll combine and we'll figure out the grand vision together. Yeah, so that's, but yeah, and the injury model, I should say, is not the one that will be pursued in the clinic. There's no way you can justify giving someone that severe injury, squirting like detergent down their their throat, but it's just currently what's working in our model.
But your assay is that injury model to measure your basal cell substrate. So isn't the mouse part? You're dependent on that being manageable or controllable in order to evaluate your culturing of these basal cells.
Yeah, I guess that's a good idea. And when we go to quantify the extent of injuries, adding in a component to look at single cell or just bulk RNA-seq immune cells and how they're responding, I think might be a good benchmark to have for some like Western analysis. That's a good idea. Yeah, Ash.
I have some questions about the confetti mice. So I noticed that you're doing 5 doses of tamoxifen.
Oh, that's what I wrote. I don't actually know how much I need.
Okay.
So that's just, I will screen up to 5 doses. In the papers I've looked at, sorry, Fisher probably was there.
No, that's okay.
Oh, okay. Just in the papers I looked at, they've generally let the sparse labeling to look at tracking digital clones, and they usually do like 1 dose of 5 mg, 0.2 mg per gram body weight. So that's just kind of what I've set so far, and I'm planning on titrating up, trying to get like the moderate recombination, but that's something I have to experimentally determine.
And do you know, have you seen in the literature when you look at these confettis with the, I guess if this report has been used, have they seen like sex-dependent recombination? That's something that we have to worry about with our confetti models. We'll see different recombination efficacies between sexes.
I have not looked into that or seen that talked about, but I will look into that. Thank you. Just yeah.
I guess I had a quick question because you were talking about clinical applications and like long term looking at the project. Would you ever think about trying to look at engraftment in a mouse model of like a chronic airway disease like CF or COPD and like not actually adding an injury, but just like, I don't know, allowing the mouse to develop and like it has this innate disrupted airway, and then you're introducing your basal cells.
What an amazing question. Trying to prove the efficacy of this like in disease correction, right? In my lab, I'm helping out. I'm someone's little helper to prepare some cells where they're going to essentially do that experiment for primary celiac dyskinesia. The Cotton Lab and like our overall group is also collaborating with a group at University of Alabama where they have a ferret program. Apparently ferrets have more human-like airways than mice. It's funny you mentioned CF because for some reason mice don't get CF. It's kind of apparently frustrating, but just what makes them better than us, I guess. But in that model, they're looking at CF ferret rescue models. So yeah, it's a good question. Someone will have an answer to that. I don't know if it'll be me, but someone will.
You'll help find the answer.
I might be a veteran. All right, thank you guys.
Hopefully it was useful, right? Have the meeting.
Sorry, right now, before all of that.
Just two.
My undergrad is coming. She messaged saying she missed the bus, so she'll be a little bit late. So then right before I left, I was like, can someone give her something to do? Because I need to prepare for it. So I'm gonna go back and talk to her, and then I'm gonna try to finish it as soon as
Lots of good answers. And if you didn't have an answer, you could just say, I'm selfish.
Yeah. But I think I have. It's a pretty good games too.
it's fine. He might not. Well, he probably was going to ask me about. He is like, can you give me the overall gist of your project in like 2 sentences?
That's possible.
Okay.
Just yesterday.
And how it is.
I realized there's this thing that happens
It's like a list of stuff to review.
automatically. It's like a small subset of HSCs garner this random mutation.
I know the PowerPoint,
It's
but which I
actually
put that
really
together
common.
after I have.
So like people are great at 60, every decade it's increased, so it goes like 5 cents. So like
Yeah,
that's
so
what I mean.
Yeah, so I'm on schedule for a
It's
couple
basically the
weeks.
Are you
And I have one like the same with 3 months.
So I'm using this chip mutation as a model of cardiovascular dissemination because it's like impossible for neurovascular.
It's so good to have mercy involved too. He's
I
so
know.
So I have my empty neuron stuff is synaptic.
Okay.
Everything else is glial.
Yeah.
Yeah.
I'm definitely a cat person.
Or monocyte. I don't know.
He might have not been.
I can't believe I just went to the shelter.
Doesn't seem like it would be related.
It was not
Were
easy.
you planning, or
No,
were
I'm
you just like, we'll see how this goes?
Yeah.
I remember
I think
going
it would
there.
be more
I'd be like, oh my gosh, I want this
relevant
small
to,
black
like,
guy called Smoke.
chromatin
And
profile
she was
and
gone,
stuff.
like, you know,
Which
it
is
was
good.
good.
The only thing I can think of is
So one of my roommates were just like,
he's like really into like ensemble related things right now because
They're like,
I mean
we have this
like,
one that